Ever wondered about the mystery behind the synthesis of proteins in our body? Today, we're diving deep into this fascinating process, which is a cornerstone of life as we know it. At the heart of this process is translation, where the genetic code is read to create proteins. But there's a key player in this process that doesn't always get the spotlight it deserves. Say hello to tRNA or Transfer RNA. This unsung hero plays an instrumental role in protein synthesis, facilitating the transfer of amino acids to the ribosome during translation. It's like a delivery service for the building blocks of proteins, ensuring the accurate and specific incorporation of amino acids into the growing chain. So if you're ready to explore the intricate world of protein synthesis and the pivotal role of tRNA, you're in the right place. Stay tuned as we unravel the role of tRNA in protein synthesis. First, let's delve into how tRNA is the key transport system for amino acids. Picture this. Each tRNA molecule is like a specialized taxi designed to pick up one specific passenger, an amino acid. This pickup happens in the bustling city of the cell, more precisely, in the cytoplasm. Now you might wonder, how does the tRNA know which amino acid to pick up? Well, the tRNA molecule has a particular anticodon sequence that corresponds to the amino acid it carries. It's like having the passenger's name written on the taxi. Once the tRNA taxi has picked up its amino acid passenger, it doesn't just hang around, it has a destination, the ribosome. This is where the magic of protein synthesis begins. The tRNA brings the amino acid to the ribosome, where it will be incorporated into the growing chain of a protein. And this isn't a one-time job for our tRNA taxi. Oh no, it's a continuous process with tRNA molecules constantly picking up and delivering amino acids to the ribosome. It's a ceaseless cycle that keeps the cell functioning and allows it to create the proteins it needs. So in essence, tRNA is like a taxi service for amino acids, ensuring they get to their destination, the ribosome. Now, how does tRNA know where to deliver these amino acids? Well, it's all down to a special region known as the anticodon. This part of the tRNA molecule has a unique sequence of 3 nucleotides, which is complementary to the codon on the messenger RNA or mRNA. During the process of translation, this anticodon region base pairs with the codon on the mRNA. This is kind of like matching puzzle pieces or finding the correct key for a lock. The anticodon and codon are a perfect fit, ensuring that the amino acid is delivered to exactly the right spot. But it's not just about delivery. This base pairing also guarantees that the right amino acid is added to the growing protein chain. Because each codon on the mRNA corresponds to a specific amino acid, the anticodon-codon pairing is like a double check, ensuring that the right building blocks are used in the right order. It's a bit like following a recipe. You wouldn't want to accidentally add salt when the recipe calls for sugar, right? And just as in cooking, getting the ingredients right is crucial for the final product. In the case of protein synthesis, this means creating functional proteins that can do their jobs properly within the cell. The tRNA in this case acts like a decoder, ensuring the correct amino acid is added according to the mRNA sequence. So how does this process facilitate protein synthesis? Well, it all boils down to a beautifully choreographed dance between the ribosome, mRNA, and tRNA. Imagine the ribosome as a tiny factory, with the mRNA serving as the blueprint for the product being made. This product is, of course, our protein. As the ribosome glides along the mRNA, reading the genetic instructions, tRNA molecules step into the spotlight. Each one is carrying a specific amino acid, like a delivery truck stocked with a particular type of material. Now remember, each tRNA molecule is uniquely equipped with an anticodon, a 3-base sequence that matches up with a specific codon or instruction on the mRNA. So, as the ribosome reads the mRNA, it's waiting for the right tRNA truck to pull up with the needed material. When a match is made, the ribosome links the amino acid carried by the tRNA to the growing polypeptide chain. This chain is the backbone of the protein being synthesized, and then the process repeats with each tRNA molecule bringing in the appropriate amino acid one by one. It's a bit like a production line, with each worker or tRNA contributing a specific part to the final product. This ensures that the correct sequence of amino acids is precisely linked together, just as the mRNA blueprint dictates. It's like a production line, with tRNA ensuring the correct sequence of amino acids is linked together. Let's talk about the importance of accuracy in this process. The specificity of tRNA for both amino acids and codons is a key player in ensuring the fidelity of translation. It's like a molecular puzzle where each piece must fit perfectly to create the right picture, or in this case, the correct protein. Each tRNA molecule is associated with a particular amino acid, and the amino acid carried by the tRNA corresponds to the anticodon sequence on the tRNA molecule. This relationship is not a coincidence but a vital part of the process. The anticodon region of tRNA base pairs with the complementary codon on the mRNA during translation. This precise pairing ensures that the correct amino acid is added to the growing protein chain according to the sequence encoded in the mRNA. But why is accuracy so important? Well, if the wrong amino acid is added, the protein may not function properly, leading to potential issues in cellular processes. So this accuracy is not just about making the right protein, it's about ensuring the smooth functioning of our cells, our organs, and ultimately our bodies. In summary, tRNA is the unsung hero in protein synthesis, serving as the crucial link between the genetic information in mRNA and the sequence of amino acids in a protein. Its vital role underlines the importance of accuracy in the complex process of protein synthesis.